Please welcome to the stage Matthew Cooley and Amanda Notke.
Okay, it's now my pleasure to call Matthew Cooley, who is a PhD student in clinical and translational science at the Mayo Clinic, and he will talk to us about the Genome-Wide association study in cholangiocarcinoma. Thank you.
All right. Well, hi, everyone. First, I'd like to thank the Cholangiocarcinoma Foundation for the invitation for us to share this information, but also for supporting this study over the last several years and allowing us to collect patient samples here at the conference. Also, as a researcher, I'd just like to thank the patients and the caregivers for sharing their stories over the last couple days. It's really motivational and inspirational for me as a researcher to hear that patient perspective and really helps me feel there's a deeper meaning to the work that our team is doing. So here today to present on the Cholangiocarcinoma Genome-Wide Association study, which everyone got the email to get their sample collected while here on site in Salt Lake City. So our agenda for this presentation, we're going to talk about what is cholangiocarcinoma, although that likely doesn't need any introduction. We'll talk about understanding the genetics of cholangiocarcinoma, describe what genome association studies are, and then introduce our International Consortium for the Genetics of Biliary Tract Cancers, long title for CCA-GWAS. So first, background on cholangiocarcinoma. So it's a cancer of the biliary tract, can generally be classified as intrahepatic or extrahepatic. We do have several well-recognized risk factors, which include cysts, stones, primary sclerosing cholangitis, liver fluke infections, but really most cases don't have a specific or known cause, and that's one of the reasons that makes early diagnosis very difficult and really highlights the importance of prevention and screening. So in terms of epidemiology, when you look at the global map, in terms of northern Europe and the US, Australia, the mortality rate is between 2 to 3 per 100,000 inhabitants. And then when you look at certain areas of Asia, it's approximately double that, where some of those known risk factors are in higher prevalence, such as liver flukes. And then you also see a good portion of the map that doesn't have any information, parts of Asia and the majority of Africa, where not much information is known about cholangiocarcinoma. So what are the risk factors for cholangiocarcinoma? As I said, it's complicated. There's some we know and a lot of things we don't know. So I think this complicated diagram is accurate. It's complicated. So there's certain things that we know have an association with cholangiocarcinoma, such as the bolded items on the slide: primary sclerosing cholangitis, inflammatory conditions of the biliary tract, liver flukes, gallstones, but also other diseases and risk factors, such as viral hepatitis, certain lifestyle components such as obesity, diabetes, hypertension, but also environment, so environmental exposures or things of that nature that can increase your risk for developing primary sclerosing cholangitis or cholangiocarcinoma. But what we're going to focus on is the host genetics for cholangiocarcinoma. So as I mentioned, the genetics of cholangiocarcinoma, they're complicated. So there's really no obvious genetic links. As far as occurrences in families, they're typically low. Most genetic links are typically attributed to the risk factors, such as risk factors for primary sclerosing cholangitis or diabetes, some of those other causes, not necessarily directly related to cholangiocarcinoma. Also making it complicated, many of the mutations that are found in cholangiocarcinoma are common cancer-causing mutations. They aren't specific to cholangiocarcinoma. They're seen in many types of cancers. Also, the genetic profile is different between intrahepatic and extrahepatic cholangiocarcinoma. So there's distinct differences between those two as well. Further complicating the matter is most of the genetic studies that have been done to date are relatively small in numbers. So the big takeaway from this slide is just showing that there's a lot of common known cancer genes, and the frequency rate in cholangiocarcinoma and gallbladder cancer. We'd think that those would be similar between cholangiocarcinoma and another similar disease such as gallbladder cancer, but it's not the case. So there's a distinct profile between those two. But again, these are just common cancer-associated genes, not specific to any one type of cancer. And then also, as I mentioned, there's distinct differences between intrahepatic and extrahepatic cholangiocarcinoma. So again, common cancer-causing mutations in oncogenes, a very distinct profile between what's observed in intrahepatic versus extrahepatic cholangiocarcinoma. So next we want to cover what the genome-wide association study is. So I think probably in a similar fashion to the east is west and west is east analogy from last night, I'll start with the largest object, which is the smallest object on this slide, a chromosome. So within our bodies we have chromosomes, which are made up of genes, which when you get smaller, they're made up of these nucleotide bases, which are kind of the building block of your genetic code. So when we find changes in your genetic code, we call these small changes are single nucleotide polymorphisms, and that's what we are trying to detect in this in our genome-wide association studies. So what are SNPs? It's the most common genetic variant or mutation, if you will. It occurs naturally and normally. They occur 1 in 1,000 nucleotides in your body. What's important to highlight is most of these SNPs, they aren't disease-causing. They occur and there's no impact because of that change. So what we want to look at is how these variants in important parts of the genes can affect a protein function, and that's when we typically see a predisposition for cancer when the mutation is at that specific location. So what we try and do in these genome-wide association studies, we sequence or genotype the DNA. We look for these SNPs. And once we sequence a large number of cholangiocarcinoma patients, we're able to compare that data to patients without cholangiocarcinoma, allowing us to identify rare variants that may be associated with this disease, which are typically less obvious in smaller study populations. So in terms of the analysis and the output, we sequence the data. We get this this long line of ATCGs. We see what's different between the affected cases and the unaffected controls, and we put together what's called a Manhattan plot, where the peak just shows us the frequency of some of these changes that were observed. And then I think I'll get into the next slides, but then we have a certain threshold which is of interest versus of significance. So now I'm going to go into kind of the background of this study, the discovery phase, and what we're working on right now, which is the expansion phase, which many of you are now going to be a part of when you donated your samples in our room. So this effort is led by Dr. Lewis Roberts at the Mayo Clinic and Dr. Catherine McGlynn, as well as Dr. Chris Amos at Baylor College of Medicine. So this is the first large-scale GWAS of cholangiocarcinoma. For the discovery phase, it was over 2,600 patients from 24 sites, primarily in North America and Europe. So in that initial data set, the majority of the participants were of European descent, a good balance of intrahepatic and extrahepatic cholangiocarcinoma, and some gallbladder cancers as well. So looking at the data, we did have some locations that met the statistical significance threshold. I think that was an HLE region, but this is just looking at our total European samples. So this is analyzing 2,400 patient cholangiocarcinoma samples. So there were some hits of interest that need further evaluation. But when we look specifically at the intrahepatic, really just some things that maybe raised our suspicion, but not necessarily to a significant state. And the same can be said with the extrahepatic samples. So the important thing with that is, we really need to expand our patient population in this study. So now what we're working on is the expansion phase, and we were recently awarded a federal grant from the NIH, which will help us sequence hopefully over 7,000 cholangiocarcinoma patient samples over the next 5 years. And we've been able to gather, I think we're at 80 some sites, maybe 85 sites now, with collaborators in over 31 countries. and a major partner in this effort is the Cholangiocarcinoma Foundation, who's been very instrumental in helping us accrue patients to participate in this study. And really the emphasis on this expansion phase is to increase the global representation and the diversity of samples in this study. So why is that important? Genetic variants are typically they can be more common in specific regions, racial or ethnic groups. And really with our first discovery phase of the project, the majority of the participants were of Northern European descent. So what we're attempting to do is really expand that to sites in Asia, Africa, South America, and additional sites across North America to hope to get a larger, a more diverse set of patient samples so we can better understand the genetics of cholangiocarcinoma. So just to highlight, here's little dots on all the countries that we so far have participating in this study. You'll see U.S. is just one dot, but we do have, I think, 30-some sites now within the U.S. that are contributing to this study, but we really expanded our efforts in Africa and Asia to really get a very diverse set of patient samples contributing to this study. So how can you participate? I think everyone got the email at the beginning of the conference, so you can complete that patient demographic form, go to room 150D, get your blood drawn, and you'll be included in that study. If you don't get a chance to do it during this conference, this is actually posted on the Cholangiocarcinoma Foundation website. You can just click that link or scan that code, submit your contact information, and we'll actually mail you a kit that you can bring to your clinic or hospital that you're seeing your provider at, and they'll collect that sample for you and ship it to us. So in terms of acknowledgements, this is our long list of sites that are currently part of our consortium focused on collecting cholangiocarcinoma samples and healthy controls over the next few years. So we're really grateful for everyone's effort to help really understand the genomics of cholangiocarcinoma. And thanks to our team at the Mayo Clinic, Baylor College of Medicine, and the NCI. So with that, I think that's it. Yeah. And I guess we'll have questions after this. Yeah, thank you.